as a SNAB, we aim to make A-level biology as simple as a cell. So today we're looking at knowing how DNA profiling is used for identification and determining genetic relationships between organisms. And I would definitely say this is one of those topics that are so frequent in past paper questions. So do keep an eye out for them. So let's look into it. So before you carry out DNA profiling, what you need to do is you need to carry out this process called polymerase chain reaction. And so basically, every individual has a unique DNA sequence. And this quality, so unless you're twins, then no, but otherwise, we all have a unique DNA sequence. And this quality, this uniqueness, can be used to develop DNA profiles. So what you can do is you isolate a sample of DNA, and through polymerase chain reaction, you can carry out, so by carrying out polymerase chain reaction, you can make multiple copies of DNA. And then you can carry a gel electrophoresis, which you will look into in a bit. You can analyze the patterns to look out for genetic relationships between whatever organism that you're investigating. So polymerase chain reaction. So DNA amplification is carried out, and this is when PCR, I will be using the acronym PCR rather than saying polymerase chain reaction each time to make things easier. So this is when PCR produces many copies of a piece of DNA. So that's DNA amplification for you. This is carried out in a PCR machine or thermal cycler, and this provides the optimal temperature for every stage and controls the duration for each stage to produce the copies of the piece of DNA. So this process needs DNA or RNA, depends on what you need, depends on context. You need primers, DNA polymerase, which we've looked into in topic 2. So if you're not familiar with DNA polymerase, do look at topic 2, which is fully uploaded by now. You also need free nucleotides and a buffer solution. So you would know why we need, you can guess why we need free nucleotides for polymerase chain reaction. And it's for the construction of new DNA or RNA strands. And you can make that connection from content we've looked into topic 2 once again. So if you're not familiar, do look at that. You also need a buffer solution. So this might be something you've looked into if you do A-level chemistry. If you don't, doesn't matter. But buffer solution is needed to ensure the pH is optimum for all reactions taking place. So buffer solution is almost able to change the pH as needed at each stage, which is why it's called buffer. So it's basically can change the pH as needed at each stage so that the whole process is happening in optimum conditions. So primers, these are short sequences of single-stranded DNA and have base sequences complementary to the 3' end of the DNA or RNA, whatever that you're copying. And this defines the region to be amplified. So basically primers, we've looked into primers in topic 3, I believe, yes, topic 3. So again, if you're not familiar with primers, do have a look at that. And this defines the region to be amplified, so it controls where the DNA polymerase needs to bind so that you can control what sections being copied. Polymerase, we have looked into this in topic 2, as I've said before, so if you want to revise, do look at it. So this enzyme is used to build new DNA or RNA strands. Taq polymerase is used in PCR, so that it does not denature at high temperature, which is needed during the initial stages of PCR reaction. So the high temperature is also needed so that annealing, which is a process once we'll look into in a bit, so that annealing does not take place at when PCR is being carried out. So what is annealing? So annealing is the joining of primers to the complementary bases at the end of the DNA fragment. So you don't want that to happen, especially for the DNA strands that you haven't even copied yet. So to prevent confusion or kind of generating the wrong copy of the DNA, you need high temperatures to prevent annealing from taking place. Stages of PCR. So you have three stages in PCR. You have denaturation, annealing, elongation. So denaturation is the DNA strand is basically heated to 95 degrees Celsius. And this is done so that it breaks the hydrogen bonds that hold the two DNA strands together. And you would know that hydrogen bonds are the bonds that hold two DNA strands together from topic 2 once again. So what is annealing? We've sort of looked into this before. So the temperature is now dropped to 5 to 60 degrees Celsius, and this ensures that annealing, because now you want annealing to happen, so it ensures that annealing takes place completely through the DNA strand. So in the initial stage, so in denaturation, you don't want annealing to take place, but at lower temperatures, it can take place because that's what you need. And then you have elongation. So during elongation, the temperature is increased to 72 degrees Celsius, and this is the optimum temperature to build the complementary strands of DNA, hence making copies of the original DNA strand. So what do you do after PCR is carried out? So the DNA is now treated with the restriction enzymes so that it can be broken into fragments of different length. So you also add a fluorescent tag so that it can be seen under UV light so that it can be studied. So DNA profiling. So now is where the actual DNA profiling bit begins. So one thing I want to make clear is that DNA profiling is not the same as PCR. This is a confusion that so many people don't wrap the head around. It's not the same thing. So DNA profiling is the actual like studying comparison of the DNA profiles you've created, and PCR is something that you do so that you can carry out DNA profiling. It's like a step towards it. So almost like if you're baking, you know, the preparation you do for the actual baking in the oven, that's like the PCR bit, and the actual baking of the cake in the oven is the DNA profiling bit. It's a bit weird analogy, but you get what I mean. So, DNA profiles of individuals can be compared to study ancestry. So, more bands that match, the closer the genetic similarity. So, it is useful to ensure biodiversity in animals and plants as closely related individuals are prevented from inbreeding. So, that's one way DNA profiling is used so that kind of closely related species do not breed. Inbreeding is avoided to reduce the risk of genetic problems possibly caused by the accumulation of recessive alleles and reduce the ability for the population to have advantageous alleles to deal with environmental changes. So especially if, you know, in zoos where maybe a certain species is borderline going into extinction, what they can do is they can carry out DNA profiling. So first they would carry a PCR and then they compare the DNA profiles DNA profiles that's created, and they can kind of crisscross the species so that more advantageous alleles are more frequent so that the species can actually survive in the long run. So yeah, that is all from Professor Snap today. This is quite a difficult topic to wrap your head around. Like PCR, the whole process might seem a lot more complicated, but if you have any questions, let me know and I will get back to them. And I would suggest going back and watching the video once again, maybe even reading things around it if you want, if that's what you're interested in, or if you just want to understand the topic better. But yeah, don't forget to like, share, and subscribe. And thank you.